Okay, welcome again, dear students, and today we will have our first lecture in biology. And we will start from very basic concepts, and then we will go further in more detailed explanation about mechanisms of life. But I would like to start from simple definition of the subject. So what is biology? Biology is science about living nature or science about life. Bios means life, logia means to study. And questions that I want to propose to all of you is why it is important part of medical education. So every time when you start to study something new, you should answer for yourself this question, why do I need this? And this will give you higher motivation for study because you will study not because it is in your curriculum and you need to pass exam, but you will study because you understand and feel why you need this subject, why you need this knowledge in your professional career. And why we need biology in medical education. First of all, human body is part of the living nature, and knowledge about living nature will provide you with very important knowledge about human body that you can use in your further study of subjects and in your professional career as medical doctors. So biology will definitely provide you with knowledge about cellular organization, about development, and about interaction of human body with surrounding world. Knowledge about cellular organization will give you understanding of mechanisms of health when you will study physiology, because physiology based on knowledge about cellular organization of the human body. Also, knowledge of cellular organization will help you understand mechanisms of disease because when you will study pathological physiology, you will study cellular mechanisms that underlie development of different diseases of the human body. And of course, cellular organization, knowledge about cellular organization is necessary to understand mechanisms of drugs, because when you will study pharmacology, you will see that a lot of drugs have their effects on the human body mediated by interaction with single cells, with certain cells with their receptors. Also, knowledge about development of human body will give you information about mechanisms of developmental disorders. Knowledge about interactions with surrounding world will help you to understand adaptations of human body to different environmental conditions and, of course, mechanisms of infectious diseases. Because, for example, parasites, they are part of surrounding environments, and in our discipline you will also study interactions of the human body with parasites that can invade human body from external world. And next important question that I would like to highlight for you is, what is the life? And luckily or unfortunately, there is no one simple definition. Life is too complex concept to be explained in one simple definition. That's why we use another way, or we use a trick to describe the life. We explain life through typical properties of living beings. So what properties common for all living organisms? First of all, it is cellular organization with tiny but extremely important exceptions that I will mention later. Then another property is sensitivity. Usually students think that sensitivity is ability to sense something from external world, and it's correct actually. However, sensitivity is not only about receiving information, it is also about possibility to somehow process this information and react in response to external stimuli or stimuli that comes from inside of the body. So sensitivity is not only ability to sense, but also ability to react on what was sensed by the organism. Growth and development, of course, reproduction, also clear, energy utilization. Every organism needs energy for maintenance of the life. And homeostasis. Homeostasis is a very important concept, and actually, I have separated several lectures about homeostasis, and if you will be interested, you can request for this lecture. But this lecture about homeostasis usually belongs to discipline of human physiology. So when you will study human physiology, I would say all discipline of human physiology will explain you how different parts of the body works in order to achieve homeostasis. Homeostasis in simple way is how organism is able to maintain its body properties in normal range of limits. How organism maintains normal temperature, normal blood pressure, normal concentration of different ions in the blood. So how organism is able to maintain its normal parameters. And problem is that when this fails, when this function is not performed properly, starts diseases. We can say that homeostasis is basis for the health, physiological basis for the health. And failure of homeostasis creates conditions for development of diseases. Of course, maybe disease will not develop immediately when homeostasis is shifted outside of the range of the normal limits, but it may create conditions that will make development of diseases more easy. Evolutionary adaptation, also very important property of the life. And now I would like to draw your attention to cellular organization of the life. And we will start from discovery of the cell. Of course, cells are so small that we cannot see them without special techniques to be used. And pioneering microscopists used their own handmade microscopes to uncover cellular level of life. Now we have extremely complicated microscopes that may occupy the whole room and costs half of the million of euro or even higher, like electron microscopes, like two photon laser scanning microscope, and so on. But at the beginning, everything was much more simple, but very important for the development of our knowledge. I would say it like this. And first microscopists, some of them were even not scientists, but just curious people who want to know a little bit more about life outside life of the world that surrounds us. So let's have a brief look on the microscope. Microscope is a special device that produces enlarged image of the object, and it consists of three main systems. One is so-called illumination system that composed of mirror and lamp. Usually, lamp is combined with condenser lens that used to focus light rays on the object. So please do not mix condenser lens with magnification lens, because magnification lens used actually to achieve magnified image of the object, but condenser lens is used simply to focus light on your object to have better illumination. So, optical system. Optical system composed of two lenses. One is objective and another ocular lens. Very easy to remember. One that is close to your eye is called ocular, and another that is close to the object that you want to study is called objective lens. Overall magnification of the microscope depends on both of them, on magnification of the objective and magnification of the ocular lens. And of course, we need focusing system. So we have to adjust plane of focus in such way that we will acquire clear, sharp vision of the object. And for this, we're using focusing system. It is mechanical change of the distance between the object and lens. And yeah, as I just told you, we use this to acquire sharp vision of the object. About those prominent person who discovered the cell. Robert Hooke proposed the term of the cell. He was a very prominent scientist of the time when he was living, and he was studying microscopic structures, and he proposed the term cell. However, at the moment when he proposed the term cell, he did not recognize that cell is a living being. He saw the structure under the microscope, but it was a static structure because what he was looking was actually not living cells, but walls of the cells. And of course, these walls of the cells did not show any signs of activity. And he did not realize that cells are actually living beings. And another person, Anton van Leeuwenhoek, he discovered that cells are actually living beings. And what is especially interesting, in contrast to Robert Hooke, who was prominent and honorable scientist, Anton van Leeuwenhoek was not a scientist. His job was to sell clothes. However, he was very curious, and he used microscope to look on the drop of water. And surprisingly, he saw very interesting life that occurs inside of one simple drop of water when he looked at it with the help of microscope. He saw a lot of, he calls them animalculus, like tiny animals who doing a lot of movements and interactions inside of small drop of water. And due to these two prominent people, Robert Hooke and Anton van Leeuwenhoek, scientific community acquired initial knowledge about cells, about microscopic dimension of living nature. And Anton van Leeuwenhoek, he recognized that these very small creatures, they are living beings. And then another prominent people, Matthias Schleiden, Theodor Schwann, and Rudolf Virchow, they analyzed information that were acquired with the help of microscopic techniques, and they created what we call cell theory. So they proposed ideas that comprised initial cell theory that actually still very relevant. Of course, now it is updated, but it is very relevant now what were proposed by these prominent people a lot of time ago. So historical cell theory composed of several concepts. Two of them were proposed by Matthias Schleiden and Theodor Schwann, and they are cell is the structural unit of life, and all living organisms comprise of one or more cells. Now it's kind of obvious for us, but for the time when these people were living, this was really important discovery, recognition that all living beings composed of cells, of one or more cells. And Rudolf Virchow added another important point to this concept that we call cell theory. And his idea was that new cells appeared from pre-existing cells through division. And this is absolutely relevant for nowadays. How modern cell theory looks like? It composed of slightly bigger number of key points, but in general, it's very similar to what was proposed before. So, cell is elementary, structural, and functional unit of life. New cells appear from pre-existing cells through division. And how this works in very simple way illustrated. Let's imagine that we have mother cell, and then mother cell divides in order to create two daughter cells. And then mother cell, of course, kind of disappeared, and instead of one mother cell, we have now two daughter cells. We will study this process in more details in our another lecture. Another important point of modern cell theory is that all cells have similar chemical compositions. So your cells, my cells, cells of some animals, they all have proteins, they all have lipids, they all have nucleic acids. So all cells have similar chemical composition. Yeah, and main organic molecules, we can say like this, are carbohydrates, nucleic acids, proteins, and lipids. Another important point of modern cell theory is that multicellular organisms have specialized forms of tissues that forms organs. So multicellular organisms, they have not absolutely identical cells throughout the whole body, but they have specialized cells that form different types of tissues that capable of performing of different functions. And different types of tissues form different organs. So as you see, specialized functional activities of the organs rely on functional specialization of the cells that compose these tissues and organs. Fascinating properties of the organs starts from special properties of the cells that compose these organs. So there is a stem cell that produce different types of so-called specialized cells, and we will discuss this concept about stem cells also during one of our seminars, because we can spend several lectures talking only about stem cells and stem cell specialization, differentiation, I would say, not specialization. Okay, so to summarize, modern cell theory tells us that cell is elementary structural and functional unit of life. New cells appear from pre-existing cells through division. All cells have similar chemical composition, and multicellular organism composed of specialized cells that form tissues and organs. Now let's go further, and since we start discuss cells, let's speak about properties of the cells. So what are properties of these elementary building blocks of our body? So cells have organized internal organization. So it is not just some homogeneous liquid solution inside, but it is actually nicely organized in order to perform a lot of activities inside of cells that are necessary for maintenance of the life of this cell and life of all cells composed of all cells that create human body, of course, will be transferred into the concept of life of the human body. And this is nice simplified picture showing that inside of the cell there is actually well organized structures. Another property of cells that is very important to keep in mind is that cells have information that is encoded in genes, and this information guides development and functional activity of the cell. So when cell is doing some type of functional activity, this happens not by random chance, but it is organized. It is organized and guided by programs. And these programs, they comes from information that is located inside of the genes of the cell. Also, important property of the cell that cells are able to produce more of themselves due to division. Of course, some highly specialized cells, they lose this property, they stop division, and they start to function in different ways. But such cells as stem cells, they maintain capability of division throughout life of the human body. And this is nice picture showing cell division. And actually, it's amazing how new microscopic techniques can show us what happens inside of the cell during different type of activity of the cells. For example, here we can see different intracellular structure during division of the cell. Another basic property of cells is that cells acquire and use energy. Also, cells carry out a lot of chemical reactions. Cells engage also in numerous mechanical activities. So they not simply staying on the one place, but some of the cells are very active. They have to migrate in order to perform their functions. But at the same time, movement of some another cells may be quite limited. Also, cells are able to respond to stimuli. As I told you before, its ability to sense and to react on what was sensed. Also, cells are able to perform self-regulation. For example, if DNA was damaged due to any reasons, there are special repair mechanisms that find mistakes in the DNA, cut out wrong portion of DNA and put inside correct portions of DNA, or if proteins, due to, for example, temperature stress, lose their correct three-dimensional organization, there are another proteins that will recognize this and will help misfolded protein to acquire normal three-dimensional organization, or if this would not be possible, destroy so-called misfolded proteins. Now let's go inside of the cells even more and speak about chemical composition of the cells. Cells have inside inorganic compounds and organic compounds. Organic compounds, what is this? I'm sure that you will study this in much more details in during chemistry, but I want to give you like brief idea of what is organic compounds already on my discipline. So organic compounds are complex molecules that have carbon chain as a molecular backbone. So this is simple illustration of it. But what you see is not yet organic molecule. It is not complete. This is only illustration of carbon chain as molecular backbone. And you see that each carbon has several, so to say, free hands, and something can be attached at each free hand of each carbon. So what are these organic compounds inside of the cell? They are lipids, proteins, nucleic acids, and carbohydrates. Let's focus a little bit on carbohydrates. What is their function? Their function is to supply energy for cellular functions. And also they are involved in synthesis of another organic molecules. They can be used for long-lasting storage of the energy. For example, accumulation of the glycogen in the liver and muscles. Imagine such situation when food is not available over more or less long period, but body needs energy, and in this case, glycogen, that is special form of accumulation of carbohydrates in the liver and also in muscles. So glycogen will be used to produce glucose. Glucose will be released from the liver to bloodstream and then taken from the bloodstream by itself. So this is example of accumulation of the energy. Glucose is kind of chemical compound that used for immediate supply with energy of different cellular function. Not exactly immediate, but it is short-term energy storage. But glycogen is a long-term energy storage. It can be stored in the liver for a long time, and then when additional amounts of glucose needed, glycogen will break and glucose will be formed and it will go through go into blood circulation. However, of course, glucose is not exactly immediate source of the energy because when glucose will go inside of the cells, it will have to be break down further, and energy that was inside of glucose will be transferred into the form of energy inside of another compound that is called adenosine triphosphate, ATP. And then ATP will serve as immediate charger with as immediate energy charger of different activities of the cells. Yeah. Examples of carbohydrates are glucose, sucrose, lactose, fructose, glycogen. So glucose, sucrose, they kind of monomers. They composed of one structure, but glycogen composed of kind of a lot of building blocks in the chain. So let's go further. Proteins. Proteins are also very important. They have numerous functions in the organisms. So they can catalyze biochemical reactions. They involved in cellular repair, growth, development. They play important role in the structure of hormones, in immune reactions. Also, they play important role in interactions between cells, in signaling between cells. And this is also very important conceptual idea that in order to create multicellular organism, it is not enough to bring a lot of cells in one place. In order to create multicellular organism, you need to establish efficient interactions between the cells. So cells will form tissue not simply because they all grouped together in one place, but because they have very efficient mechanisms for interaction with each other. Also, proteins play important role in cellular movements. They play important role in transport of substances across plasma membrane. And in some not favorable circumstances, I would say like this, proteins also can be used as a source of the energy. So when glycogen storage depleted, and if food is not available for a very long period of time, as last reserve of energy, organism can start to destroy some proteins and use them in order to acquire additional energy. Energy for critically necessary cellular functions. Of course, it's not good for organism to destroy its own proteins. However, in order to keep running critical process in cell, organism can start to destroy proteins that are involved in not critical processes that are running inside of the cells. Next one that we will discuss is nucleic acid. Nucleic acids, they are used for storage of hereditary information, and this is information that guides development and functional activity of the cells. They used for storage of this information and also for transfer of this information from one generation to another generation. And this is relevant when we consider human body, and this is also relevant when we consider single cell. Because as you remember during our previous slides, I showed you how mother cell divided and two new daughter cells created. And this is also kind of one generation and another generation, but in terms of the cells. And nucleic acids were used in order to transfer hereditary information from mother cell to daughter cells. And of course, when we will speak about different generations in terms of the whole human body, nucleic acids used to transfer genetic information from parents to children. And also nucleic acids used for expression of hereditary information that guide development and activity of the organism. So three main points that you should remember about genetic information and nucleic acids. Nucleic acids used to store hereditary information, to transfer it between generations, and also nucleic acids used for expression of hereditary information. For example, let's imagine we have data server somewhere in one room, and it is used for safe storage of information. And you need this information in another room in order to use it somehow. And for this, it is not enough to store, because if you are in the room number two, you cannot simply use information that is stored in the room number one. So you need somehow to take this information from there and to use it. And for this usage of hereditary information, also use nucleic acids. So we have different types of nucleic acids, some of them used for storage and transfer between generations, and another type of nucleic acids is used for expression of hereditary information. Lipids. What do we know about the lipids? Lipids play important role in the structure of cell membrane. So first idea that should come to your mind when you hear lipids is that they are major component of the plasma membrane. Of course, lipids have also another functions, but mostly in our discipline, we will focus your attention to role of lipids in structure of plasma membrane. And plasma membrane, understanding of plasma membrane also very important for, I would say, all your future medical career, because interaction of the cells that are one of functional basis of the health, and disturbances in interactions of the cells represent one of functional basis of disease development. So these interactions between the cells occurs via plasma membranes. Also, mechanisms of drugs, pharmacological mechanisms. Pharmacological drugs They have influence on cells that are usually mediated by plasma membranes. So plasma membrane is critical player in maintenance of the health. It is critical player in disease development, and it is very important player in mechanisms of actions of drugs. These three concepts that you will use during all your medical career. And now we make foundation of, make a ground knowledge that you can use to make your knowledge later more advanced in other disciplines. But even longest journey started from first small steps, and I hope that today we are making together this first small step in your understanding of cellular structure, of how cell is organized, and how plasma membrane is organized. So today we're making very important step of your very long and I hope very successful journey in your professional career. Lipids also play important role in the structure of hormones and vitamins. So as I told you just recently, lipids not only involved in construction of plasma membrane, they also have another important functions like structural components of hormones and vitamins that are very important. Also, they used to store big amount of energy over a long period of time. So lipids, they also can be used for long-lasting storage of the energy. Uh, and good example of it is adipose tissues. So adipose tissues is very important because it provides us with possibility for long-lasting storage of the energy. And also, due to recent advantage, regenerative medicine start to use adipose tissues as source of stem cells that represent very big hope of medical science in terms of development of new treatment strategies for different types of diseases. Oxidation of lipids produce more energy than oxidation of carbohydrates. So as you can see, that lipids can provide us with big amount of energy. And also lipids can be used as a source of metabolic water. So what is metabolic water? Metabolic water is water that is synthesized inside of the cells of our body. Not the one that we drink, but the one that is synthesized in our own cells. And now let's focus a little bit more on cell membranes. Those ones that I tell you will be very important for all your medical career. Cytoplasmic membrane. What are their function? What are the function of cytoplasmic membrane? I would say like this. And these functions are very important. So first of all, and what is very obvious, cytoplasmic membrane separates cell from extracellular environment. But it is not only about isolation. This is also about interaction with external world. For example, your skin. Your skin is used to separate your body from external environment, but also you acquire a lot of information through the skin, through your sensory system that is associated with the skin. And also you acquire a lot of components that can somehow transfer through the skin. So skin is not only a mechanical barrier, skin is also a tissue that perform very important function of interaction between your body and external world. Something similar may something similar may be presented as analogy when we're talking about cytoplasmic membrane. Cytoplasmic membrane from one side separate cell from extracellular environment, and this is very important to keep uh, intracellular structures safe. But in order to run a lot of processes inside of the cell constantly needs to acquire some additional energy, additional structural materials from extracellular space, and at the same time, cell have to release some compounds from inside to the outside. So cell have to be in interaction with extracellular space, and this is also done with the help of cytoplasmic membrane. Cytoplasmic membrane involved in transfer materials between cell and its environment. Also, cytoplasmic membrane involved in informational interaction between different cells. As I told you before, different cells have to communicate with each other in order to form tissue, and this is done with the help of cell membrane. One cell produces chemical compound that is used as chemical information signal for another cell, and this chemical information signal released from other cell transported through extracellular space and reach another cell, and then it have to be it has to be sensed by specialized structure on cell membrane of another cell. So cells they use to release information, cell membrane used to release information signal and to receive information signal. And this is very, very important concept that is used in physiology, pathophysiology, pharmacology, concept of information signaling between different cells that occurs via cell membrane. Also, cytoplasmic membrane involved in mechanical interaction between cells. So, some tissues require tight and reliable physical contact between different cells. So cells have kind of stick to each other and keep this mechanical contact very reliable. And this also occurs with the help of cell membrane. And if some cell have to migrate to different locations, this cellular movement also also achieved with the help of cell membrane. Not also, but cell membrane is important player in cell movement. And what are chemical components of cell membrane? So after we received initial ideas about what cell membrane are doing, what cell membranes are doing, we have to understand, at least in simple general way at the beginning, what is the structure of the cell membrane. And I would suggest you this stepwise approach for each topic that you learn further. At the beginning, you should ask yourself simple questions about general concepts. And when you find answer on these simple questions of general concepts, you start to ask next more detailed questions. For example, simple question, what what are the functional role of what are the functions of cell membrane? And then when you understand this, okay, I understand. Now I asking another question. Okay, what is the structure of cell membrane? And then what are detailed mechanisms of each function? So from simple question, you go inside further and further and further. But typical mistake of many students is try to study without systematization of their knowledge. They try simply to memorize as much pieces of information as possible, and they jumping from initial concepts to very tiny details, and then it becomes a mess in their memory without structured information. And this is one of the ways to memorize, but it is not the best way, because without a systematic picture in your brain, big amount of details soon will be lost. A good way to acquire long-living knowledge is to create a system. And this system always starts from simple concepts, and then you attach additional detailed points on initially constructed simple concept of the subject that you study. So let's go further and speak now about more details of cytoplasmic membrane, and we will speak about components of cell membranes. Principal components of cell membranes are phospholipids, proteins, and carbohydrates. And the major component of cytoplasmic membrane is represented by phospholipids. So here you see, I would not say simplified, but rather nicely detailed uh, illustration of cell membrane. And you see that there are a lot of structures painted in red color, not so many structures painted in blue, and just few structures painted in green color. So those guys that are painted in red color, they are phospholipids. And you see how a lot of them are presented in this picture. In blue color, highlighted proteins. And you see that their number is not so high as number of phospholipids. In green color, highlighted carbohydrates, and their number even less than number of proteins. But despite of limited number of different components, they all play very important role. So without proteins, plasma membrane will not be able to perform its functions. Without carbohydrates, also a lot of critically important functions of plasma membrane would not be possible. So integrity of all components are necessary to provide plasma membrane with possibility to perform all its functions. And once again, I would like that you remember from this lecture that plasma membrane is not only about separation, but it is about interaction of cell with extracellular space. And this concept of interaction involves everything that we were talking about in relation to cytoplasmic membrane. It involves transfer of chemical compounds inside, outside. It involves informational interaction between cells. It involves mechanical movement. So all functions that we highlighted for cytoplasmic membrane can be packed in one concept of interaction between cell and extracellular environment. What proteins present inside of the cell membrane? We have two possibilities to describe these proteins. One possibility is to describe proteins located inside of cell membrane according to their location. Kind of very simple approach. And another approach to describe proteins that are located in cell membrane is according to their functions. According to location, we distinguish integral protein, peripheral proteins, and surface proteins. According to their function, we distinguish channel proteins, carrier proteins, and pumps. Also, very important proteins that are located in cell membrane called receptors. Another important proteins, they are called adhesion proteins. Proteins that are responsible for physical contact between different cells. And now let's switch to another topic that also related to the cells. Let's speak about different classes of cells. Of course, if you will look inside of the human body, you will see very much, sorry, you will see a lot of different types of cells, a lot, and we probably will need maybe 1 week or 1 month in order to describe different types of cells of the body, but actually even not months, but the whole year. And there is a discipline, histology, that doing this job, describing different types of cells in the human body. But there is an approach that can categorize all cells into two big categories. And these categories are eukaryotic cells and prokaryotic cells. So all cells in the human body, despite their very big number of differences, they all belong to one class of the cell that we call eukaryotic cells. And there is another class of the cells that also include in itself a lot of different cells, but in general, there is another big class that is called prokaryotic cells. So, in very general overview, we can distinguish, very general overview, all cells in our nature we can classify in two groups. And these groups are prokaryotes or prokaryotic cells and eukaryotes, eukaryotic cells. And all cells in the human body, they belong to eukaryotic cells. Of course, when we're talking about cells that belongs to our body, if we are not taking into consideration some parasites that can be located inside of our body, because these guys can be prokaryotic cells. And what are the difference between these two very big categories, very big group of cells, prokaryotes and eukaryotes? Prokaryotes, their major difference from eukaryotes is that they do not have nucleus. So simple idea, if nucleus is not present, we call the cells prokaryotes. If nucleus is present, we call the cells eukaryotes. And once again, all cells that belong to human body, and you will spend a lot of time to study different types of these human body cells, they all belong to eukaryotes, cells that have nucleus. But presence of the nucleus being very obvious is not the only one I would say, point of differentiation between prokaryotes and eukaryotes. Prokaryotes, they also have simple organization of genetic material, while eukaryotes, they have much more complex organization of genetic material. And as my mentor said long time ago when we were talking about I would say, weakness and strengths of certain method, she told me that it's not reasonable to say that something is weakness or the strength, but more correctly to say this is a property. Why? Because same property, depending on situation, can be weakness or can be advantage. So we see that prokaryotes, they have simple organization of genetic material, but eukaryotes more complex. And this gives eukaryotes kind of advantage in terms of number of different reactions that they can do. So they can do more tricky reactions on situations that coming from external world and situations that maybe happen inside of the cells. So they are more advanced in terms of adaptive reaction. However, a high level of complexity require high level of organization and more time for processing information, more time in order to achieve some action. While simplified organization gives to the cells, I would say, limited number of actions, but they can do them more fast. And in some situations, this speed of events can be benefits. Also, prokaryotes, they do not have specialized intracellular organelles, while eukaryotes have specialized intracellular organelles. Intracellular transport of prokaryotes is not structured, while transport of eukaryotes is highly organized. And prokaryotes, they have simple and fast division, while eukaryotes, they use division by mitosis and meiosis. And this is one of example how sometimes simplification may lead to some advantage, but not to disadvantage, because these cells they able to divide more fast, and they can expand their numbers during limited time. So they take less time for division than cells of eukaryotes, because in order to divide, eukaryotic cell have to organize a lot of things inside before and during division. Prokaryotic cells can do this more simple. Why this is important? Because some of prokaryotic cells are agents of infectious diseases. And you can imagine that increasing number of agents of infectious diseases in the human body will lead to increase of severity of disease. And this is what you have to keep in mind, that these guys, they expand their number fast, and that's why in some situations of infectious diseases, your decision about therapy of diseases also have to be done fast. Because if you will wait for too long time, these guys will multiply themselves to such amount of cells that their summarized action will be very severe, and they may lead to very severe consequences for human body. So these guys, they are fast in cell division, and they can. This is one additional properties that have to be taken into consideration when you making decision about the treatment. And also prokaryotes, they not have sexual reproduction, while eukaryotes have sexual reproduction. Well, actually, prokaryotes have something looking similar to sexual reproduction, but not true sexual reproduction. Because during true sexual reproduction, uh, two cells fuse together in order to create new cell. What prokaryotes are doing, they able to fuse two cells together, but this fusion is temporarily, it is not complete, and after this fusion, there will be no new cell created. So what doing prokaryotes? Two cells come together, they establish contact between each other, and then they share some portions of genetic material with each other, then they become separated and continue separated living. So they not fuse completely, they not create new cells due to this fusion. They simply comes very close, establish contact, exchange with genetic information, and then they separate. What eukaryotes doing? They produce specialized germ cells that fuse together and create one new cell. Okay, now let's speak briefly about organelles of eukaryotic cells. And as you can see in this beautiful picture, there is a one big nice organelle inside and a lot of not so big organelles, but they are also very important. This big beautiful organelle is nucleus, that is kind of information center of the cell. It safely store genetic material inside in the form of nucleic acids. Ribosomes, tiny, tiny yellow dots that you can see here, they used to produce new proteins. And the plasmic reticulum that you see here in green color is used for the, I would say, processing, but this now may be a complicated concept for you. Uh, let's imagine such situation. You created a protein with the help of ribosome, but this protein is not yet ready uh, to perform its functions because this protein did not have necessary three-dimensional organization. And the plasmic reticulum is place where freshly synthesized or newly synthesized protein will acquire its three-dimensional organization that will be necessary for its further functional activity. So, in the plasmic reticulum that is associated with ribosomes used for three for construction of special three-dimensional organization of the proteins. And also the plasmic reticulum responsible for another functions that you will study in more details when you will read the textbook. For example, calcium can be stored in specialized endoplasmic reticulum and then used in so-called cellular signaling. Complex Golgi that is, I will show you now, moment. Yeah, so this is complex Golgi. It is also very important, actually all is very important. So complex Golgi is kind of post station. So let's imagine that you created something and then you want to transfer this product that you have created to another person. You go with this product to post station, you pack this product into a package, and then you deliver it to the place where it will be needed. So when proteins are synthesized with the help of ribosomes, they transferred into endoplasmatic reticulum here in order to acquire special three-dimensional organization. And then proteins have to be packed and transported further to the place where they are needed. And this packaging of the protein products into transport vesicles occurs inside of the complex Golgi. Centrosome, this structure is necessary for mitosis, for cell division. It is used to construct special tubules that will be used in transport of genetic information uh, inside of the cell. So far, I think it's enough, and we will speak about this in more details on the lecture about cell division. So centrosome involved in mechanical movement of genetic material during cell division. Also, another very, sorry, I also always say important, but actually everything is important. So another important organelle is mitochondria, kind of powerhouse of the cell. It is used for transformation of the energy from long-term storage to readily available chemical form of the energy. For example, uh, or even I would say from short-term storage to readily available form storage of the energy. For example, when glucose enters into the cell, glucose is kind of short-term storage chemical form of the energy. It is transferred into mitochondria, and there glucose is being processed in such way that it breaks down and energy is released. And at the same time, this energy will be transferred into the form of adenosine triphosphate, and adenosine triphosphate will be used as chemical form of immediately available energy. So glucose energy that is located inside of glucose cannot be used immediately by the cell in order to uh, supply different types of cell activity with the energy. In order to supply different types of cell activity, energy should be transformed from the form of glucose into the form of adenosine triphosphate. And then adenosine triphosphate can be used for immediate energy supply of different events that occurs in the cell. And this transfer of energy from the glucose, from the form of glucose into the form of adenosine triphosphate occurs inside of the mitochondria. So once again, glucose is kind of short-term energy storage, while adenosine triphosphate is special compound that is used for immediately available energy supply of different activities running inside of the cell. And this transfer occurs in mitochondria. The structures, so-called lysosomes, they are membrane vesicles full of with proteins, full of with enzymes that can be used for digestion of different organic compounds. And also these enzymes can be used for destruction of some pathogenic bacteria. For example, immune cells of the body, macrophages, they are able to eat pathogenic bacteria, and then when pathogenic bacteria appears inside of this macrophage, vesicles containing digestive enzyme will come in contact with this pathogenic bacteria and should destroy this pathogenic bacteria. And if, due to some genetic disorders, these vesicles will not function correctly, those pathogenic bacteria that located inside of the macrophages will have possibility to survive. And if they will survive, of course, efficiency of immune system goes down. So this is some simple example how functional activity of lysosomes is necessary not only for simple maintenance of the life of the cell, but is important, for example, in immune function. And at the end of our lecture, I want to discuss levels of organization of life. So living nature is complex system and it can be categorized on different levels. And one level that we consider to be initial level of organization of life is level of cells or cellular level. Then next level of complexity of living nature represented by tissues. Cells organized together with possibility for interaction with each other will form tissues. Tissues organized together will form organs. And with each step in this complexity level, we will see acquisition of new functions. So organs can perform functions that actually not possible for each separated tissues that included in this organ. Tissue can perform more complex function than every single cell. Next level of complexity or organization of life is level of the organisms. Next level is population level, then comes ecosystem level and biosphere level. Another question that I want to discuss you at the end of our lecture today is classification of living organisms. So I want that you have kind of overview of organisms that create our nature. So you know that all cells can be divided for prokaryotes and eukaryotes. And prokaryotic organisms, they can be further subdivided for several domains. So they are eubacteria and archaebacteria. Eubacteria and archaebacteria. Eukaryotic organisms can be subdivided for different kingdoms. Kingdom of protists, and protists are single cellular eukaryotic organisms. So they are organisms that composed of single cell that have nucleus. And some of important parasites that can lead to diseases are represented by these guys, by protists. And we will study them in more details during section of parasitology. Another kingdom is fungi, another plants, and another animals. And also, I want to draw your attention to one kind of special case in the classification of living organisms, because all these organisms that belongs to prokaryotes and eukaryotes, they all can be considered together as cellular living systems. And at the same time, in our nature, there are non-cellular systems. And they kind of on the border between living nature and not living nature, because you remember, cells considered to be elementary unit of life. And if there is no cell, there should be no life. However, there are special guys that we call viruses that are not cells. They not have cellular organization, but they are very active players in living nature. So they start to behave themselves as living being when they integrate into the cell. Outside of the cell, they not able to reproduce themselves. Outside of the cell, they not able to perform activities that considered to be basic properties of life. But once they integrate inside of the cell, they start to behave as living beings. So all living beings in our nature can be categorized to non-cellular and cellular. Cellular further categorized to prokaryotes and eukaryotes, and then each of these groups, eukaryotes and prokaryotes, can be categorized further. So, dear students, that's it for today. I hope that this lecture was interesting for you, and this is only first initial step in very long and I hope interesting journey in biology.